Right, so thank you very much. Thanks for inviting us to speak today. So the title of our talk is Vegetable Crop Improvement in a Changing Climate. So I'll be looking first of all at something called the VeGIN project, and then, as Lauren said, Jim is going to talk about some of the work that's spun off of that, and also how that has influenced really where we're taking VeGIN forward in the future. So it all ties together. So what does VeGIN mean? So this project is called the Vegetable Genetic Improvement Network. And as the name suggests, we are trying to improve crop traits within a range of different vegetable crops because of some of the challenges that are present currently and potentially in the future going forwards with commercial vegetable production. So this project is a collaboration between ourselves at Harper Adams, but also the University of Warwick. So it's led overall by Guy Barker at Warwick, but there's a number of colleagues, Lauren and Rosemary included, who work on this project with us at Warwick, and I'll explain how that works a little more in a second. But in the latest round of VeGIN, we've also got the involvement of the UK Met Office, which is a great extra addition that will allow us to really find out some more on the climate side of things in this project. So VeGIN's funded by Defra. It's gone through a number of iterations over the number of years. It's been going for about 10 or 15 years now, and we're very grateful to them for that funding support. It's actually one of several different similar projects looking at different crops. These are all Defra funded. There's 5 different ones. So as well as the vegetable crops aspect that we look on, there are also analogous genetic improvement networks or GINs for wheat, oilseed, soft fruit and pulse crops. But within VeGIN, how we divide our work up is based around the different organizations that are involved. So University of Warwick handle the biotic trait side of things, so looking at pest and disease resistance in these vegetable crops. At Harper Adams, we focus on the abiotic side of things, so environmental stresses, nutrient depletion. I said the Met Office have recently come on board in terms of climate modeling, which will hopefully try and tie things together, particularly with some of the abiotic work. But one of the key strengths of VeGIN, the involvement of a large body of industry stakeholders. That's predominantly crop breeders, but also producers. And they have the ability through regular interaction to help steer the research focus of the project. And that helps us maintain a direction that's as commercially relevant and therefore as commercially useful as possible, because we get feedback on maybe the particular traits we're looking at, the particular crop species that are in most need of assistance in terms of identifying new trait resilience currently or in the future. So what kind of work do we do in VeGIN? Well, you could broadly overall describe it as what we call pre-breeding research. This is pre-commercial, essentially trait identification. Can we take lots of different germplasm, lots of different crop varieties, and identify traits of interest, maybe resistance to a particular disease, pest, or environmental stress? And then can we also try and elucidate something about the underlying mechanisms that might confer that particular trait, so maybe candidate gene identification? The aim being is that we can then either provide physical material or data regarding things like quantitative trait loci, candidate gene information, gene expression, to crop breeders to allow them to hopefully improve the durability and the resistance of the varieties that they create in the future. In order to do this, we need a source of germplasm. So we need somewhere to get our plant material from. And we're fortunate that we're able to make use of the UK Vegetable Gene Bank, which is based at Wellesbourne as part of Warwick. And there they have a very large number of accessions of a wide range of vegetable crops. And VeGIN has been able to take subsets of that material and develop populations that we can put into our phenotyping systems, identify the traits we need, and move forward from there. So which crops do we study? One of the challenges with VeGIN, which differentiates it from some of the other GINs, is that we have a whole range of different crops that we could possibly choose from. To date, we've tend to focus on the 4 main UK vegetable crops in terms of commercial value and on area grown. So those would be the vegetable brassicas, so things like broccoli, cabbage, cauliflower, but also lettuce, carrot and onion. But now as VeGIN moves forward, we're expanding our remit and including additional things like leafy herbs, such as coriander and other root crops, including parsnip. So just an example of just a selection of some of the kind of traits that we've looked at and are continuing to look at within VeGIN. With lettuce, for example, disease side of things, lettuce wilt and root rot caused by Fusarium oxysporum, or with pests, lettuce root aphid. At Harper, we've looked at drought, waterlogging, and nutrition issues with lettuce, and I'll talk a bit more about that later. With vegetable brassicas, we've looked at traits including resistance to clubroot, cabbage root fly, and again, the abiotic stressors. Some examples from carrot would include cavity spot, carrot red leaf virus, and again, abiotic work. And the abiotic work has continued with onions as well, and I'll mention that briefly a little later. So first of all, I'm going to talk a little bit about some of the abiotic stress resilience work that we've done in VeGIN. I'll talk first of all about some of the vegetable brassica work that we've looked at. So why do we want to do this? Well, crop growing is stressful, not just for the people who are trying to do the growing, but also for the crops themselves. Now, I won't talk about the biotic side of stress today, but I'll be focusing on the abiotic side of things. And with this, we could be referring to the availability and distribution of nutrients in the soil, or we could be talking about environmental effects. So this could be water availability or excess, so drought and waterlogging, fluctuations in temperature, so excessive heat or cold, or maybe soil quality issues such as increased salinity. When we're thinking about the effects of the environment on crops, we need to think about, are we thinking about the wide scale, or are we thinking on a local basis? And are we thinking about short or long-term growth conditions? Are we thinking about changes in climate that might occur gradually over a number of years, or are we talking about day-to-day weather patterns? So within VeGIN, we've taken a particular approach. So we're all aware that with climate change, we need to potentially adapt the crops we're growing to cope better with the kind of conditions we expect to see in the future. Thereby, we need to develop more environmentally resilient crops. Now, as part of climate modeling, we found that we get a predicted increase in the frequency of extreme weather events. So it might be fluctuations in temperature, fluctuations in precipitation, and thereby we would expect to see increased day-to-day weather variability. That might lead itself to producing short-term, i.e. transient periods of abiotic stress in a crop, whereby it might encounter particularly hot or cold weather for a couple of days before returning to more typical ambient conditions, or the same with water, for example, a heavy storm causing waterlogging over a short period of time. Therefore, as well as producing crops that can cope with chronic long-term changes in growth conditions, we also need to develop those that are better able to cope with short-term fluctuations and thereby short-term periods of environmental stress. But in studying this in vegetable crop production in the UK, we also need to acknowledge the way vegetable crops are grown and produced within this country. So for many fresh produce crops, things like brassicas and lettuce, they're actually originally sown and raised indoors under cover in large-scale glasshouses, and they're grown until you generate a seedling with about 3 or 4 true leaves. That seedling, which is growing in a small block of usually something like a peat-based or other medium substrate, as you can see in the picture on the right, together that seedling and the block are referred to as transplants. And you can see in the background the trays of transplants ready to go out into the field. So once those plants have been hardened off, they'll be taken out and the entire block, root system and seedling will be planted into the field as a single unit. So we need to acknowledge that when we try and design our phenotyping assays and we choose our selection of material and the types of treatment we impose when working to get a most commercially relevant assay system within VeGIN and therefore the most commercially useful data. I've mentioned already transient abiotic stress. Now, this point of transplantation where these young plants are going out into the field, before they can probably establish in the field, thereby getting their root system out of the block that they were previously grown in and into the field soil, being able to extract nutrition and water from there, until they can probably establish, they're vulnerable to fluctuations in environmental conditions. So this is an important stage in the crop production cycle of these crops where that risk of negative effects due to abiotic stress is key. And that's why we focused on this particular stage for the imposition of our stress treatments to make them more commercially relevant, but also with the added benefit that we're able to work with smaller plants and increase the throughput in terms of either number of plants or number of different varieties that we're able to screen. So we're very fortunate being able to work with the UK Vegetable Gene Bank, but that also presents itself with a bit of a challenge in that we have the luxury of many different accessions of germplasm available to us, more than we could feasibly phenotype. So we need a way of narrowing down the amount of material that we need to phenotype, but still maintaining as much of that diversity as we possibly can. And there's been a number of different crop populations developed within VeGIN that try to extract the most variability, maintain the most diversity, but by reducing the number of lines that are screened. Some examples of this might be what we refer to as diversity fixed foundation sets, or DFFSs. This is where we take maybe only about 100 different lines, but we try to, in that selection, maintain the maximal amount of diversity in terms of genetic background, morphology, geographical origin, and so on, to try and make a balance between feasibility in terms of phenotyping, so getting down to a number of lines that we can actually phenotype with a suitable amount of replication, but trying to maintain as much diversity as we possibly can. These diversity fixed foundation sets, in these, the lines tend to have been genetically fixed, so they're homozygous, often done through a process of called double haploid generation, which means that for every accession, each seed is essentially genetically identical, which makes it much easier to work with these populations, makes it easier to get data where we can interpret more easily what's going on. So how have we done this? So having selected our germplasm population, we need a way of assessing the effect of our transient stress that we impose on any one plant. And the way we've chosen to do this is effectively with a mirrored approach, where for each particular accession, we will have a control plant or group of control plants, and we will have a number of plants that have been stressed of that same accession. In order to monitor the effect of stress, we're not recording at the point of stress imposition, but instead we allow them a period of recovery, and then we compare the growth in terms of simple characterization like fresh weight, dry weight, overall plant size, leaf number, just very basic morphology parameters. And we look at that of the stress plant compared to the control. And that allows us to generate things like a relative fresh weight. So 100% would be no different between the stress and the control plant, and a relative fresh weight of 50% would mean the stress plant was half the size of the control plant. And this allows us to easily rank and compare the different accessions, because between the accessions there can be quite a difference in their appearance and their overall growth rate, and this allows us to account for that. So the way we've done this with the brassicas is we tend to germinate the lines initially in commercial module trays. So these are called 3, 4, 5 module trays because of the number of cells in each one. These are the kind of things that brassicas will be propagated in commercially under cover. When they get to around the transplantation stage, so 3 or 4 true leaves, we then select plants at the closest size that we can across the whole lines and between the controls and the treated plants, and we impose our transient stress. This is an example of plants being subjected to a heat and drought shock within a phytotron. So they start off by not looking too good. By the end of the treatment, they're looking really quite sad, but we're able to fortunately recover them from that. So because we don't have space in the rhizotron for this amount of plants, they go back in the glasshouse, we water them up, pot them on, and we let them recover for a couple of weeks. Now, although they've recovered, those plants that underwent the stress treatment never properly recover in some of the lines. So we still see a knock-on effect on things like fresh and dry weight 2 weeks later, and this is what we measure in a lot of these assays. The kind of things we might see could be something like this, where we've got a control plant belonging to a particular accession, a drought plant where we can see the drought has had a slightly negative effect in terms of leaf size, the overall plant size, and then waterlogging, you can see, is quite dramatically reduced the size of the plant. So this transient stress, and you can see how in the assays, the stress treatments are really quite short. Even when the plants are returned to optimal conditions afterwards, that short period of stress has had a knock-on effect, and that plant hasn't recovered. But it allows us to compare between different lines. Occasionally, we might see something like this, where the stress actually stimulates a response whereby the stress plant is larger than the control plant. This is not something you'd expect, and we don't see it very often, but it does crop up occasionally. What we're looking for really is a plant where the stress plant and the control are no different. Thereby, having applied a stress to it, we've not had a negative effect. So the less decline, the better. So if we do this across an entire population, this is the kind of data we get. So this is an example where we've droughted some brassicas belonging to the Diversity Fixed Foundation set for Brassica oleracea, the vegetable crop species, and we're looking at the relative fresh weight of the stress plant compared to the control. So the ones on the left, where the relative fresh weight is the lowest, the stress plant is smaller than the untreated plant. So they've been knocked back a lot by the drought. The ones we're really interested in are the ones on the right. Here, where the relative fresh weight is around 100%, there's very little difference between a plant that's been stressed and one that hasn't. So these are the ones that are the most resilient and therefore potentially the ones of most interest going forwards. What we've done is we've tested a lot of individual stresses, first of all, against the different oleracea varieties. So we applied a drought stress or a flood stress and so on. And we wanted to know, is there a correlation between how these plants respond to one stress and to another? And overall, it would appear not. So we can't predict the response of a plant to, say, heat stress by knowing what it does with salinity or with cold. So there's overall no correlation. Occasionally, we might see a line, for example, this one here, where it appears to be susceptible to all of the single stresses, or this one here that appears to be all resilient. Interestingly, these are both broccolis. But overall, we cannot say that there is really much correlation between the response to the different stresses. There's one small exception to that, and that is in the response of leaf generation to extremes of temperature. When we look at the plant leaf number in these plants, whether we've imposed a heat stress or a cold stress, the extreme lines, that is the ones that did the best or did the worst, are largely the same whether we're looking at heat stress or cold stress. So that suggests that a line that's able to produce leaves after it's been subjected to heat stress, it's also able to do the same if it's had a cold stress. So perhaps there is some sort of shared extreme temperature response here. I say this is the exception rather than the rule. Overall, we don't see a correlation of responses to different stresses. Now, at this point, we've only looked at single stresses. In reality, in the field, you're likely to encounter multiple stresses at once. For example, if you've got very high temperatures, it's likely that you might be encountering drought at the same time. So combined stresses is another thing we've been looking at in VeGIN throughout the project. I mentioned earlier that within Brassica oleracea, we've got a number of different crop types. So this single plant species is responsible for the whole range of different food sources you see at the bottom there. So things like kale, cauliflower, cabbage, kohlrabi, broccoli, and so on. So we wanted to know, is the response that we see in terms of stress resilience linked in any way to the type of crop that we're growing? And again, we found no significant difference in stress response between different crop types. And in a way, that's good, because if we found that only broccoli, for example, was showing responses that were maybe drought resilient and all the cabbages or the kales were really suffering, that doesn't bode so well for trying to breed that resistance into those crop types that are not doing so well. But given that we found resilience across a number of different crop morphotypes, as you might want to describe them, that's a promising result going forwards, and that we're not tied to just one particular crop type within the species. So just to wrap on up on this point in the brassicas so far, we don't seem to see differences between crop types, but that's a good thing because we can potentially breed resistant or tolerance to abiotic stress across different types of brassicas. We've also done some work looking at wild relatives of crops to see if there's anything in there, any genetic material that can help us that may have been lost through the processes of domestication and generation of cultivars. So within VeGIN, we have another of these diversity fixed foundation sets, and that's called the Brassica C genome. So this was established from 89 different wild relative Brassica accessions from 14 different species. But in order to genetically fix them and overcome the issues of self-incompatibility that you get with a lot of these brassicas, they were crossed with a self-compatible oleracea line. So the lines that we've got here are essentially hybrids. And again, if we subject these to different stresses, whether we're combining two stresses together like drought and heat, or single stresses on their own, flooding and salinity, we see significant differences across the population, some doing better than others, which is good. So far, so good. So which wild relative might be causing this, maybe providing us with some beneficial traits? Well, that's harder to say because we didn't see any significant relationship between which wild species was present as one of the parents of the line and the response we were seeing. Now, should also come with the caveat that the replication numbers, the number of lines with these different parents is quite low, as you can see at the bottom. So this warrants further investigation. But so far, it doesn't appear that any particular wild relative is conferring resistance to these stresses. So just to conclude the brassica work, it seems that different brassicas respond in different ways to different stresses, and there's no correlation on the whole between the different stress types and the way the lines respond. But we are finding stress resilience in multiple crop types. So how about carrots and onions? Now, carrots and onions are a bit harder to work with than brassicas. With the brassicas, we were able to generate these double haploid homozygous lines, get nicely uniform seed, and enable us to get data that we can usually make more use of statistically and practically. However, it's not easy to generate homozygous lines with carrot and onion. And instead, we create what are called half-Sib populations. So this is through a process of what we call limited open pollination. So we have a limited number of parent individuals, and we use those to generate the seeds to form each accession. So the uniformity is not to the same degree, but we can still use these populations to screen for responses to different stresses. So here's an example looking at onions, and we've plotted their response to drought, that's the red dots, and flooding, that's the blue dots. And what we're showing here is for each of the different varieties what we call the effect size. So the lower the number on the effect size, the more the negative effect the stress has had. So what we're looking for in a good variety is one that has a positive value for both blue dots and red dots, for both flooding and drought. And we can use this approach to pick out those that are either unaffected by the stress or maybe even slightly increased, and we can eliminate those that are impacted most dramatically in a negative way. And we can do exactly the same thing with carrots using the same approach. So this is data for a range of carrot varieties from the gene bank. Again, looking at their drought and flood responses. So they're ranked by flood, and you can see the ones at the top have the best ability to deal with flooding, but how they deal with drought shows basically no correlation with their ability to deal with flooding. We just have to pick out those lines that appear to do best in both circumstances. So finally, I'm just going to do a quick few slides talking about some of the work we've looked at regarding nutritional stress in VeGIN. So today, so far, I've talked mainly about responses to things like temperature variability, water availability, but now we're going to look at one aspect, looking at nutrient deficiency. So in this case, we're looking at a problem called tip burn in lettuce. So that's shown in the image on the right. So this is a problem that's thought to be due to insufficient calcium delivery to the leaf tips. So the tissues there start to break down and die. We get leaf discoloration, and obviously this affects marketability. The problem with this condition is that it's affected by many environmental factors. So it can be hard to predict. One of the main ones being humidity. So it's thought that high humidity around the leaf slows the transpiration stream through the plant, and therefore it means it reduces delivery of calcium to the leaf margins and encourages this destruction of the tissue. But it's still not 100% certain what exactly is going on inside these plants. So in order to screen some of the VeGIN diversity set for tip burn resilience, first of all, we needed to develop an assay in which we could do that. So we needed a phenotyping method to allow us to reproducibly obtain tip burn so that we could then try and identify those lines that were more resilient. So we settled on a hydroponic system here. So this is kind of like a small scale deep water hydroponic system. So we've got lettuce plants that are generated in blocks, first of all, very similar to those transplants, and then transplanted into perlite in a pot and submerged in aerated nutrient solution. We were able to use this to fairly rapidly, within about 3 or 4 weeks, obtain a wide range of tip-burn symptom severity across the different populations we were looking at. We didn't want to have an assay where everything was severely affected, but neither did we want an assay where everything was unaffected, just like we would have in the drought or other assays. In terms of tip-burn symptom scoring, we just used a simple scoring scale from mild to moderate to severe to monitor the symptoms we were getting as a rapid way to determine susceptibility across a large number of plants. So we used a number of different populations in here. We had a lettuce diversity fixed foundation set, which is analogous to the ones that we've used with brassicas, but in this case it had about 70 varieties of the commercial lettuce, like Tuca sativa, and then 16 wild relative species lines. We've also used a mapping population generated from 2 commercial cultivars, so that's a repeatedly inbred mapping population in order to get what we call recombinant inbred lines that are segregating for traits from the 2 parents, which are called Saladin and Iceberg. So going back to the diversity set, if we look at all the different varieties, we get a whole range of tip-burn symptoms, which is what we were looking for. We have those that are most resilient with the lowest tip-burn, so these are the ones that we want, and then the ones with the highest tip-burn scores are the ones that are most susceptible, so the ones that are most problematic. But thankfully, we've got a good range from one to the other and everything in between. So it's the resilient lines that are of most interest. We also did some very crude morphology scoring on these plants to try and get a bit of an idea about what's going on in terms of what kind of morphological traits do they have that might be conferring resistance to tip-burn. And we found that the lines that have wider leaves or rounded leaves, or ones that were curled as opposed to flat, or that had some red pigmentation as opposed to all green, were significantly more susceptible to tip-burn. So a plant that had intermediate or narrow leaves that were serrated or flat or entirely green without any red pigmentation tended to score lower in terms of tip-burn, which is a good thing. However, we also need to look at the different species that were in this population, because that might help explain some of the things that I've just mentioned. So when we look at the different species that are present, but we need to bear in mind that there are a lot more sativa accessions than there are the others, we find that the sativa accessions, so the cultivated species, have significantly higher tip burn than the wild relatives. So far, this looks promising that the wild relatives have much less severe symptoms. So what might be causing this? So if we go from tip burn score on the left, where the wild species, Saligna, Seriola, and Verosa, have a lot lower tip burn, to the percent dry weight in the middle, it turns out that they have a higher percent dry weight than the cultivated species, sativa. So could this be partially responsible for the difference we're seeing? But the other thing we did was we did a crude scoring of head density. So some lettuces, like iceberg varieties, they ball up. They make a very dense ball of leaves. Others are more open, things like butterhead lettuces, and we get everything in between. So using a crude head density score, we also did that for the 4 different species present, and we find that the cultivated varieties have a much higher head density. And that might be a bit clearer when I show you what these kind of plants look like. So a cultivated lettuce on the left, like you might be familiar with, very high density of leaves. That's what they've been bred for. We want a lettuce that has a lot of leaf material on a single plant for something especially like an iceberg, a high density of leaf matter, those crunchy, well-packed leaves. A wild lettuce, on the other hand, looks more like something on the right. And in fact, it looks a bit more like a thistle than a lettuce. You can see that it's a much more open, much less dense plant. Now, that's not what we want commercially, but what that would do is improve the airflow around the leaves. If that airflow is improved, then that will drive the transpiration stream and potentially encourage the distribution of calcium to the leaf margins. So this morphological feature of these wild lettuce species may be the reason why they're getting lower tip burn. The problem is, the reason they're getting lower tip burn is because of traits that are not agronomically favorable. So this low head density, for example. So we therefore need to go back to just the cultivated varieties and see if there's anything in there that isn't a morphological trait that can help us with tip burn resistance. Unfortunately, we do find some resilient lines even within the cultivated species, which is more promising in this case than going for the wild relatives. We can also try and find a bit about what's going on underneath the trait by screening the mapping population. So this is the Saladin times iceberg mapping population. And again, we get some resilient lines, some susceptible, and some in the middle. Using this, we were able to identify a number of small effect QTLs, but nothing that seemed to have a big contribution to the trait that we were looking for. This is not surprising given the amount of environmental influence on tip burn development. So it's hard to get nice QTLs that contribute a high proportion to the trait you're seeing when you've got such high environmental influence upon the trait you're scoring. So it's hard work, but we did manage to find a number of small effect QTLs here. So where do we go next for VeGIN? Most of what I've talked to you about so far has been developing phenotyping assays, screening material, identifying germplasm that looks promising for downstream breeding approaches. But we want to find out more about why, what is going on with this material. So recently, a new 5-year VeGIN program has been funded starting last year. And within this, we're going to try and target more some of the underlying genetic components for some of these traits. We're also going to branch out into looking at more of the role of the roots. So far, we've predominantly focused on shoot material and how that links to abiotic stress traits, but we're going to focus more on root system architecture in brassica, carrot, and onion. Jim is going to talk about some of this work in lettuce as well shortly, and how that might link to abiotic stress resilience and their ability and efficiency of nutrient use. We're also going to start looking at the links between abiotic and biotic traits. So, for example, the interaction between the root system architecture of a lettuce and its resistance to root aphids. But in terms of identifying what's going on underneath these traits, we are going to be following a pipeline of transcriptomics, targeted QRT-PCR, and progressing forward to gene editing to try and identify candidate genes and elucidate what function they are playing in terms of these resistance traits. And then that provides more markers, more genetic information for breeders going forwards. So I'd just like to acknowledge everyone who's worked as part of the VeGIN team at Harper and also at Warwick. So thank you very much for everyone who's been involved with VeGIN over the years, to Defra for funding, and from a huge body of stakeholders that provide an invaluable part of the scheme. So I'm going to hand you over to my colleague Jim Monaghan now, who's going to talk about some of the projects that have arisen from VeGIN. So thank you very much.
Right, thank you very much, Andrew. Lauren, can you see my screen and can you hear me fine?
Yes to both, thank you very much.
Right, okay. So I shall carry on Andrew's excellent introduction there into what's been going on in VeGIN over the last few years. And I want to just use two examples where we've taken these VeGIN resources and then we've started to move along this pipeline that Andrew just outlined there. So I'm going to look at using VeGIN resources to study lettuce rooting traits. So the first piece of work was undertaken by a PhD student, Justin Roberts, who's now a breeder in a UK breeding business. And the aim of this was to look at this rapid rooting phenotype. So the title of his PhD was Identifying a Rapid Rooting Phenotype for the Improvement of Lettuce Transplant Establishment. And you'll see if you look in the list, it was collaborative work between Harper Adams and Nottingham University, but also Syngenta supported this project. So why was he interested in lettuce rooting at the transplant stage? Well, lettuce is manually harvested still, though that is changing and we're seeing mechanized systems coming into play now. But crop uniformity is really important and that's not going to change. So people grow lettuce from transplants in the field because you have larger plants growing away quickly. You don't have any variation to do with emergence or seed germination, etc. But you do have some issues with establishment time as a source of variation. And many factors influence transplant establishment. There could be the soil, it could be how well the block has been placed. And then, as Andrew was talking previously, you have these transient stresses at transplanting when you have this block, which is has all the roots that are sustaining the plants, and yet you've not properly engaged with the soil environment around you. On top of it, you can also get some pruning of the root system. And this image up here, you'll see where here we've only put one lettuce seed in the block, but you can see how far the lettuce roots have moved around the tray. And when these individual blocks are removed, that you're actually tearing roots, and that can have an impact. Now, the population we used, Andrew's talked about, is the diversity fixed foundation set in lettuce. And this is 96 lines that were chosen to represent the total genetic resources available. So rather than having multiple lines which are very closely related, it gives a wide spread. And so that had the sativa types and then also Cereola, Saligna and Verosa included in the population. Another VeGIN resource that we used in this project was a mapping population of rills from a Saladin iceberg cross. And these were actually generated originally by Dave Pink for downy mildew studies. But we found that they segregate for a number of other things, post-harvest discoloration, and we've done some work on that in the past and published it. And in this case, for lettuce rooting traits. Now, the rill population, whilst they come from two parents, Saladin and iceberg, is quite a variable collection. These are the rills that I've got here, not the diversity set. And you'll see you've got different forms, you've got different colourings. These are much more paler, these are darker greens. And so it's quite a variable phenotype. And intriguingly, when you look at the parents, you have a variety called Saladin, which is an iceberg type, and a variety called iceberg, which isn't an iceberg type, it's actually a Batavian or a Batavian type. But if you look at the progeny within this population, around about 50-50 split falls into a COS type morphotype and an iceberg type morphotype. So this is the diversity set, sorry, this is the rill mapping population that we used. So Justin grew his lettuce seeds on a 2D growth pouch assay that had been established at Nottingham. And essentially you'll see up here a seed is being placed on this paper, on this rooting paper, and then a black polythene sheet was put over the top. The pouches were then put in these racks, and we were able to do 300 seedlings per rack. And because we did these in a controlled environment, we didn't have to put all the seeds in at the same time. We put the seeds in at the same stage of germination. And I'll show you that in a slide or so. And we would let these grow down the paper, and 2 weeks later, we would remove the pouches, remove the black polythene, and then image the root system. And you can see a really nice example. Here's your primary root, and here are these lateral roots coming off. Now, to deal with the fact that some of these lines germinated earlier than others, what Justin developed was a protocol where he germinated all the seeds in this manner on Petri dishes. When the radical had emerged and you started to see root hairs, that would be the stage he would transfer the seeds into the assay. So we would measure from germination till the end of the study rather than it being a calendar date. And he put them just 2 to 5 millimeters below the surface, again, to get optimized emergence. And so you don't want the seeds sitting too high because of issues with too much light and also the risk of them drying out. So the sort of data we got out of this assay, here's the diversity fixed foundation set. And on the x-axis and the y-axis, we have different measures. So the x-axis, we have the mean total lateral root length. So this would have been collected from, we were using ImageJ at the time, and then the mean number of lateral roots. And you see there's a relationship where over here we've got one accession which has got very long lateral root length and a large number of lateral roots. So we're able to phenotype the diversity set in this manner, but we could also phenotype, in this case, commercial lines down here. So these were commercial lines which are anonymized. And then we also have the parents of the real population, iceberg and Saladin. We have quite a nice segregation there. And here's two of the diversity set, just to give you an indication. That's Cacaticia white and Schladder that were on the previous slide. But these, what does this actually mean? Well, they look like this. So we'd have, here's our Cacaticia white, which has got an awful lot of rooting going on, and the numbers get less. But more interestingly here is the commercial lines, and they're showing that actually we have purposely or inadvertently selected for lines where there are a few lateral roots and not much strength in the primary root. And if you contrast that up here with this extreme phenotype we have in Cacaticia white. So we could see variation in phenotype across both the diversity set and the mapping population. And here's the rills in here. So here's the parents. So you can see there's a little bit of transgressive segregation, but most of them fit quite nicely between the two parents. So working with the rills, we were able to then develop a QTL map, and we could see that across the 9 linkage group or chromosomes in lettuce, that there were a number of QTL. Linkage group 2 was very active, and also if you look, you've got some QTL in linkage group 4, 5, 7, 8 and 9, so quite dispersed. So we ended up with 12 statistical QTL identified for these rooting traits. So we concluded we can breed for rapid rooting. That's great. We've shown genetic variation in the diversity set, and then we've also been able to get into the rills to try to get these QTL. And we published this paper a few years ago. So the question is, where do we go next? And that's a nice, by the way, a wild lettuce that Andrew and I saw in the Czech Republic, growing out of the pavement, whilst we were at a conference on lettuce genetics. But we must get out more. So where are we next? So we've got a new PhD with Cara Wharton, and this is collaborative work with Miriam Gifford at Warwick. And this is taking Justin's work onto the next step. So we've got the transcriptome from the parents, and we've got the transcriptome from the extreme rill lines. So Cara has been able to go hunting for genes to see, with some differential expression work, to see whether she can identify potential genes of interest that we could then move forward within breeding programs. So she started off with 401 differentially expressed genes in the parent line, so that's Saladin and Iceberg. She then had a different data set to look at the differentially expressed genes in the extreme rills that Justin had identified previously, and we got down to 42 genes. And of those, 21 genes were expressed in the same direction in both populations, so the parents and the rills. And from those, Cara's then been working on looking at genes of relevance in the literature where these genes have been shown or reported to have some impact on rooting traits. And Cara's moved away from the pouch assays that Justin was using and is now moving into more realistic substrates. So here's some work that Cara's been doing using rhizotrons. And you'll see we've got Seriola, Cacaticia white, Schladder, Iceberg. And she's imaging, quite a challenging job in this environment, but she's imaging these and then getting quantitative data on root traits, as well as getting leaf biomass data as well. And what you'll immediately notice is these are bigger plants than, of course, than the seedling work that Justin was doing previously. So we're moving that forward, and we're seeing similar patterns of phenotype. Not completely a one-to-one relationship, but our extremes within the extreme rapid rooters and the slow rooters, we're also seeing extreme rapid rooters and slow rooters at this phenotyping assay. So where do we go next? Well, we've got genes that we want to know more about. So the first thing we need to do is validate these genes. So Cara's taking 3 approaches. She's identified and sourced tDNA Arabidopsis lines with the candidate genes knocked out. And so she's phenotyping these in a novel array that she's developed. Rather than full rhizotrons, she's actually using root trainers as her root trainertrons. And that paper will be coming out in Plant Methods, has just been accepted last week. So that will detail the approach she's doing to get low cost root trait assaying. The other approach is that we're having a go at CRISPR. The pipeline's underway, and we've sequenced the genes and the guides have been developed. And so that's taking place at the Elizabeth Creek facility at Warwick. So that's great that we can work with that facility, all within VeGIN, of course. And so that would allow her to validate again whether her genes of interest are playing a key role. And finally, Cara's come up with a novel way of silencing the genes. On those rooting pouches, we're in a position where we can actually apply RNAi using a system developed by Mahmut Tor at Worcester. And so she is just in the process of sorting out the process, optimizing that for this approach. So that's a whistle-stop tour on how we've taken VeGIN resources and we've gone along the journey from phenotype now towards identifying gene function and target genes for breeders. So I just conclude. This is there's Andrew up there. So this is some of the group who are working with us in the area of phenotyping and vegetable crops. And over here is Miriam Gifford and Miriam Gifford's group. And again, Miriam is collaborating with us on Cara's project. So I will stop there and hand back to Lauren.
Thank you very much to both of you for really interesting presentations. If you're happy to, are you okay if we open the floor to questions? Because there's already a couple appearing in the chat. So if any of you do have any questions, please do put them in the chat or raise your hand and we can invite you to ask your question in person. First question, I think this was during your talk, Andrew. Are productivity and stress tolerance negatively correlated?
That's a great question. The short answer is I can't say that they are, because we haven't taken a lot of these varieties in the population through to a field trial to look at productivity. It'd be an interesting thing to do with some of the extreme lines, but also a lot of the varieties that we've been using are not necessarily modern commercial varieties, and that's something that it would be interesting to look at. You might expect that that there is a negative correlation because maybe plants that are optimizing, maybe baseline stress responses are impacted if that stress period doesn't occur. But then again, if it does, maybe they will do better. So maybe you would see a positive correlation. So yeah, it's a great question. I can't give a definitive answer, I'm afraid, but it'd be something very interesting to look at.
Brilliant, thank you. Okay, and I think we've got a question for you now, Jim. Are the results of your root assay compromised by the use of block transplants in commercial production?
Thanks, Ian. Great question and a very good question because of the root pruning aspect. Justin, in his work, and this is unpublished, but it's in his thesis, he actually looked at that and he established that rapid rooting phenotype as identified at the seedling stage was maintained if you then went into a block and you pruned all the roots off the block. You still got rapid root growth out of the block. So I suppose it's a yes, they are, and but the extent that they are would seem to be less concerning.
Brilliant. Thank you very much. Okay, anybody else got any questions? Please do post in the chat or raise your hand. A question from me, this is a slightly random one, but in terms of the blocking, the industry is looking to move away from kind of traditional peat blocking. What kind of impact do you think that might kind of have in the kind of realm of research that you've both been doing?
So that's really interesting. There's change going in two directions at the moment. We've seen people move from the traditional block tray. So Andrew has been doing work in module trays. So they're brassica module trays. They're lettuce blocking. So they're different things, but the same. They're both very restricted root space for young plants. You're seeing the industry going two ways. One is they've been moving towards something called plant tape, particularly in lettuces, though that direction has done a bit of a handbrake turn recently. But that is where you're growing in much smaller volumes of peat, and you're putting your seedlings into the ground at an earlier stage. You might have one or two true leaves emerged. There are issues with that when it gets into the field in terms of consistency. And so now what we're seeing, along with the push to get away from peat, and peat is a great product for making blocks, is we're seeing people move towards sort of Japanese pots, maybe like an Ellie pot style system, which is a bigger substrate volume, but it's actually holding it together. So we are seeing changes in transplant production, but as I come back, the work that we've done so far in the area, it would suggest that the phenotype is maintained at an early stage, regardless of the module size.
That's brilliant, and I guess very encouraging considering the long time it takes to be breeding plants. So with changes in kind of industry, we need to ensure that what reaches the growers is consistent with these changes. Okay, I'm not seeing any more questions coming through. Let me just double check there's no hands up. No, I can't see anything else coming through. So in that case, I think we'll say a huge thank you to Jim and Andrew and end this meeting for today.